Hello, IB Biology class. This is Ms. Shealy with your next installment of your video notes. Don't forget that while you're watching your notes, listening to me talk, you need to take notes and write down any questions so that when we go over these topics in class, you have those questions ready. All right, today's topic, we're still in Chapter 5, which is Evolution and Biodiversity, and we are looking at Section 3 of Chapter 5, which is Classification and Biodiversity. The first thing that we need to remember is the idea of the binomial nomenclature, or two-named naming system. We've learned this in introductory biology as well. So if we look at Homo sapiens, which is us, the first name is the genus and the second name is the species. That genus is always capital letter and the species is always lowercase lettered, and we're always writing it underlined. If we're handwriting or if we're typing it, it's in italics. The genus name can be abbreviated if you have previously used that information in your text. But how did this binomial nomenclature idea start? It was developed by Linnaeus, who was a Swedish botanist and taxonomist. It's still used today as the basis for classification and the naming of all new species, and this makes it consistent amongst scientists around the world. It's based on physical traits of the species, and most of his classified groups are still accurate today, and he was way ahead of his time. Let's watch a little clip.
Carl Linnaeus was born in the southern Swedish province of Småland in 1707 and is considered to be one of the most influential scientists of his time. His life's work was to develop and refine a way to group life on Earth, or to classify it. So straightforward was his system, it is still used by scientists and understood by people across the world today. His father, Niels Ingmarsson Linnaeus, was both an avid gardener and a Lutheran pastor, and Carl showed a deep love of plants and a fascination with their names from a very early age. While studying medicine at both the universities of Lund and the University of Uppsala in Sweden, he devoted a large amount of his time to the study of plants, and in 1732 he mounted a botanical expedition to Lapland. Over 5 months he travelled some 3,000 miles, collecting biological specimens and taking notes. In 1734 he mounted another expedition to central Sweden.
He was an empiricist. He didn't believe things that he hadn't seen with his own eyes. He was very reluctant to take on trust something that somebody had written about.
In 1735, Linnaeus moved to the Netherlands, and it was in that same year that he published his most well-known work, the Systema Naturae, or the System of Nature. In it, he outlined a new system for the naming of all living things.
There are two main things that Carl Linnaeus tends to be known for. The one is as far as his classification systems, which at the time were really rather revolutionary, particularly in the plant kingdom, rather less so amongst the animals. And the second thing is his introduction of a new naming system using a genus name and a species name, the so-called binomial system. Now, of the two, it's the latter, it's the naming system that is his primary legacy.
This item is the first edition of Systema Naturae, the one where Linnaeus actually outlines his sexual system for classifying plants, basically by the number of stamens and the arrangement of the reproductive organs. So this is the first time it's appeared in print, and so that's why it's important. You can see it's very large, and I tend to explain it as being the 18th century version of an Excel spreadsheet, because you can see it's very table-like. By the time the 10th edition appeared in 1758, this had shrunk from this really large size to a much more sort of portable thing, but it had gone from only 11 pages to 2 fat volumes.
Returning to Sweden in 1738, he practiced medicine, specialising in the treatment of syphilis, and lectured in Stockholm before being awarded a professorship at Uppsala University in 1741. Throughout his life, Linnaeus showed an interest in nature and the systems which govern it. He wrote not just about classification, but also about ecology, how organisms interact with their environment. He explored food chains and even defined the concept of race, dividing humans into 4 groups: Americanus, Asiaticus, Africanus, and Europeanus. Forced to retire from teaching in 1774 by a stroke, Linnaeus suffered a further stroke and eventually died in 1778. Linnaeus was driven by a lust for all nature and the desire to understand and classify it. His legacy remains and is used by the many dedicated scientists today who are driven by that same desire.
All right, so let's review the hierarchy of taxa. Taxonomy is the practice and the science of classification, as you just saw in the video about Carl Linnaeus. He was one of the first to come up with this idea of taxonomy. There are 7 levels of classification, going from the largest, the domain, to the smallest, the species. As we move from kingdom to phylum all the way to species, the organisms share more and more characteristics. This system is going to allow us to group organisms whilst also assigning unique species names, and it's very helpful in identifying and naming new species. So it works for not just naming existing organisms, but also being able to add in new species as they're discovered. So just a quick review of what a species is. We've actually already gotten into this. This is not more later. A species is a group of organisms which can interbreed and produce fertile offspring. Species are named with a genus and then a species. So this zebra longwing has a genus and a species that's specific to it. The smallest taxonomic group is the species, but there are sometimes many subspecies that are recognized. Subspecies might potentially interbreed if a barrier or other challenge was removed, such as distance. And you can see subspecies of the white peacock butterfly. One is this one here on the left is found in Puerto Rico, and this one here is found in Jamaica. So if the distance and the water between the islands was removed, they could actually interbreed, but there's enough difference between them to make them subspecies. All right, so there are 3 domains. The 3 domains are bacteria, archaea, and eukarya. Looking at these 3 domains and looking at 4 different features and how they are present or absent or how they appear in these domains, it can easily be shown why the bacteria and the archaea became their own domain and split off from the original kingdom Monera. So when we're looking at histones, remember those are the proteins that are associated with eukaryote DNA. They are wrapped around and found, they're called nucleosomes. They are absent in bacteria, but there is a protein that's similar to histones that's bound to DNA in archaea, and of course they are present in eukaryote. Introns are the sections of the DNA or the mRNA that stay in the nucleus and do not exit to code for proteins to go to the ribosomes to make proteins. They are rare or absent in bacteria. They are actually present in some genes in archaea, and they are frequent in eukaryote. The cell walls in bacteria are made of peptidoglycan. Remember that peptidoglycan, it has to do with whether it's going to stain positive or negative in a gram stain. So that's the difference between your bacteria and your archaea. So bacteria has peptidoglycan, archaea does not. Eukarya, our cell wall is not made of peptidoglycan, but it's also not always present. The most common cell wall is going to be in plant cells, and that is typically made of cellulose. The cell membrane, all cell membranes are made of glycerol ester lipids in all 3. They all have unbranched side chains, but both the bacteria and the eukarya use the D form of glycerol, and the archaea is the L form of glycerol. So remember, that has to do with the arrangement of the atoms in the glycerol molecule, and we looked at that much earlier in the year. Then here you can see a nice phylogenetic tree of life for the 3 domains. All right, so also in this section, we're talking about naming, we're talking about binomial nomenclature, classification, so of course we need to talk about the dichotomous key. Dichotomous key is a tool that allows the user to determine the identity of a certain animal, whether it be a tree, a flower, an animal, or any other living object. Dichotomous actually means divided into two parts. Therefore, dichotomous keys always give two choices for each step. And they can be in a branched form like this, or they can be in a list form. You'll find the list form if you have a lot of options that you need to get through. The branching can just become too complex. So here, all of our samples have feathers. So yes or no. Do they have feathers? Yes. And then you have a question, does it swim? Yes, then it's a duck. No, then it's a hen. If it does not have feathers, then the next question is, does it have legs? Yes, it has legs, then it's a lizard. No, it does not have legs, then it's a snake. So that's a very simple dichotomous key. Here, you've got one whether it has a backbone or does not have a backbone. If it does have a backbone, then you go to question 2. If it does not have a backbone, then you go to question 5. This one we might be more familiar with if we've used them in secondary high school before. All right, so here's the plant kingdom. The plant kingdom is divided into several different types of plants. You have vascular plants and non-vascular plants. Within the vascular plants, you have spermatophytes. They produce seeds which contain sperm, or you have follicophytes, and then the seed-producing spermatophytes are angiospermatophytes or gymnospermatophytes, and then angiospermatophytes, or the flowering plants, can be monocots or dicots, or monocotyledons or dicotyledons. You're going to focus more on this angiospermatophytes next year in the second level of IB Bio. This year we're going to focus on some of these differences here. So we're basically going to look at some differences between basic non-vascular plants. You have aquatic algae and mosses. We are not going to focus on the aquatic algae. We're going to focus on these last 4. We're going to look at the Bryophytes, the mosses, the Follicophytes, the ferns, the Coniferophytes, the pines, such as pines, and then the Angiospermatophyta, which is the flowering plants. So we're going to look at some specific differences between these plants. All right, so the first thing we're looking at is their vegetative organs. So Bryophytes have no true roots. Instead, they have something called a rhizoid. Here is a Bryophyta down here. The rhizoids, they do look like roots. They are really just there for anchoring and a little bit of absorption, but more just for anchoring. Follicophytes, Coniferophytes, and Angiospermatophytes, so ferns, conifers, and flowering plants, all have root stems and leaves. So they have roots for anchoring and absorbing, stems for traveling, that allowing that vascular tissue to be present, and then leaves for the photosynthesis to happen. All right, so that takes us to our next column, which is vascular tissue. Vascular tissue has to do with xylem and phloem. Xylem is typically water and mineral travel, whereas phloem is going to be water and food travel. So water does flow through both. Bryophytes don't have either xylem or phloem, but xylem and phloem are both present in Follicophytes, Coniferophytes, and Angiospermatophytes. So because they have root stems and leaves, they are also going to have xylem and phloem. All right, the next thing we're going to have look at is something called cambium. Cambium is a layer of tissues that's going to continue to produce more tissue, and it's going to be just underneath the bark. So you're not going to find a cambium in either a Bryophyta or a Follicophyta, but you will see it in a Coniferophyta and Angiospermatophyta. So in evergreen trees and even in flowering plants where you have more of a woody stem, you're going to find a cambium. So that's going to be in your woody plants. Pollen. Pollen is, of course, our sperm of our plants, our gamete, male gamete. There is no pollen in Bryophytes or Follicophytes. The pollen in Coniferophytes is produced in male cones, and the pollen produced in Angiospermatophyta is produced in flowers, specifically in the anthers. So you can look at the picture down here and see the anther here. All right, the opposite gamete, your ovules producing the egg gametes, you're not going to have any ovules in Birophytus or Phyllocophytus. You will have ovules in Coniferophytus produced in female cones, and in Angiospermophyta, they are enclosed inside of the ovaries within the flower itself. Now, since Birophyta and Phyllocophyta don't have pollen and ovules, it should not be a surprise that there are no seeds either for either of these two things. Seeds in Coniferophyta are produced and dispersed as well as they are in Angiospermatophyta. They are produced and dispersed within fruits for Angiospermatophyta, whether that be the flower or the fruit itself. There are no fruits in Coniferophytus, Phyllocophytus, or Birophytus. All right, now we're going to talk about the animal kingdom. You can see that the animal kingdom is first divided into two sections, the invertebrates and the vertebrates. So we're going to start with focusing on invertebrates, and then we'll move into vertebrates in a little bit. We are going to look at poriferas, which are sponges, cnidarias, which are typically jellyfish, anemones, Platyethtomies, which are flatworms, annelidas, which are segmented worms, molluscas, snails, slugs, and octopus, and arthropodas, which are your insects, spiders, crustaceans, millipedes, things like that. All right, so the first thing we're going to look at is whether they have a mouth or an anus. So there is no mouth or an anus for a porifera. There is a mouth only for a cnidaria and a Platyethtomies, and there is a mouth and an anus for the mollusca, annelida, and arthropoda. As far as symmetry is concerned, first of all, you need to be aware of three major types of symmetry. So, of course, first you have no symmetry. You have bilateral, where if you segmented it right down the middle, it would be the exact same on each side. And then radial, which no matter how you separate it equally in half, it is the same. So porifera has no symmetry. Remember, those are sponges. No matter how you were to lay that line of symmetry, you're not going to find any mirrored image. Cnidarias typically are radial, that's your jellyfish, and then all the rest are typically bilateral symmetry. All right, so the skeleton aspect of these guys, porifera have internal spicules, which are kind of like skeletal needles, but no typical skeleton that you're familiar with. Cnidaria have a soft skeleton, so not a typical external or internal hard skeleton that you're used to, although there is an exception in that hard corals do secrete calcium bicarbonate, which will form that hardness around the coral to protect it. Platyethtomies have a soft skeleton, so essentially no skeleton. Mollusca have a shell made of that same calcium bicarbonate, and then annelida have an internal cavity that is filled with fluid that's underneath the pressure. And then arthropoda have their external skeleton, which is made of plates of chitin. So remember, these are all invertebrates, so they have no internal skeleton such as a chordate would that we're familiar with. So that is all the vertebrate side. All right, so other external recognition features. So porifera have many pores because they are filter feeders, and they are very, very varied in shape, which is why they have no symmetry. Cnidaria typically have tentacles that are arranged in rings around the mouth, and they also have stinging cells. Platyethtomies have a flat or thin body that's in the shape of a ribbon. They have no blood system and no system for gas exchange. Mollusca have a mantle, which is just a large fold in the body wall, and that mantle will secrete the pieces of the shell to form the shell around them. And they also have a hard, rasping radula that is used for feeding. So they do have one little hard area that is used to help them feed. Annelida are bodies made of many ring-shaped segments, and those segments often have bristles, and their blood vessels are also visible. And then arthropoda are segmented bodies and legs with joints between the sections. So just to give you an idea, the last two had segments, and I just wanted to remind you visually that segmented is going to mean like many sections, and then no segments will be an entire, no sections, no division. All right, so now we're going to look at vertebrates, and we're not going to look at all of these vertebrates, but we are going to look at some big ones within this dichotomous key. All right, so we're going to look at 5 main types of chordates. So we're going to look at bony ray finned fish, amphibians, reptiles, birds, and mammals. We're just looking at some major differences between the two. So first, looking at their outer surface. Bony ray-finned fish have scales, which are actually bony plates embedded in the skin. Amphibians have a soft, moist skin that is permeable to water and gases. Reptiles, in difference, have an impermeable skin, and it's actually covered in scales that are made of keratin. Birds have a skin that's covered with feathers that are made of keratin, and mammals have skin that has follicles that contain hair made of keratin. So a lot of keratin there in the last 3. And then how do these guys exchange gases? Well, bony ray-finned fish have gills. Amphibians have simple lungs with small folds. Reptiles have developed lungs with extensive folding. Birds have lungs with parabronchial tubes and air sacs. And then mammals have lungs with alveoli, ribs, and a diaphragm. So starting off kind of small, and you can see the progression of So starting off kind of small, and then you can see the progression of complicatedness to lead to greater and greater surface area for gas exchange. All right. The next 2 columns go together. They're appendages and kind of in addition to that. So first of all, bony ray-finned fish have no actual limbs, but all amphibians, reptiles, birds, and mammals all are tetrapods. So they have 4 limbs, and each of those limbs are pentadactyl, where you have an ending in 5 digits. Now, even though bony ray-finned fish don't have limbs, they do have fins that are supported by rays. The amphibians, although they are tetrapods, they have 4 legs when they're an adult. They do not have 4 legs when they are younger. Reptiles have 4 legs in most species throughout their entire life. Birds have 2 legs and 2 wings, although they are tetrapods. And mammals have 4 legs, but sometimes could be 2 legs and the other 2 are either wings or arms. All right, how are the differences in their development? Well, bony ray-finned fish actually remain in water for their entire life cycle. Amphibians start in the water in their larval stage, but then they are on land for their adult stage. Reptiles and birds both lay eggs. The female lays eggs, but reptiles are going to lay a soft shell, whereas birds are going to lay a hard shell. And then, of course, mammals give birth to live young and then feed those live young with milk from mammary glands. This column is just kind of miscellaneous information that is important to differentiate between the different phylum of chordates. You have bony ray-finned fish have a swim bladder that's going to help them with buoyancy since they do remain in the water for their life cycle. Amphibians have eggs that are coated in a protective jelly since their larval stage does start in the water. Reptiles have teeth of all the same type. Sorry, that's a typo. Their teeth should be all the same type. Birds have a beak but no teeth, and mammals have teeth of different types, and there is a living core to those teeth. And then the last thing that we're going to look at is body temperature. Bony ray-finned fish, amphibians, and reptiles, none of those 3 maintain their body temperature, but both birds and mammals do maintain a constant body temperature. That's all for today's notes. I hope you jotted down some great new information. There is a little bit of memorization with this and understanding those differences between those 3 different groups. Please bring your questions to class, and I'll see you soon. Thanks.